Bismillahirrahmanirrahim. Today the topic of my presentation is identifying role of genetic and environmental factors in disease causes. I am Dr. Abhay Nazan and presenting under kind supervision of Professor Dr. Sairaj. Learning outline. Genetic epidemiology, different studies, applying molecular technology, assembly studies, and sequencing technology. Learning outcome. Concept of genetic epidemiology, role of genetic and environmental factors in disease causation, and concept of genetic technology. Now, what is genetic epidemiology? New medical discipline that seeks to understand how genetic factors interact with the environment in context of disease in population. Areas of study include the cause of inherited disease and its distribution and control. Traditional genetics focus primarily on single gene traits that follow the transmission pattern. Carrier screening followed by appropriate genetic testing can reduce the burden of disease. Twin studies. Studies of twins have been of great value in identifying the relative contributions of genetic and environmental factors to the causation of disease. Age distribution in published childhood leukemia in twins. That is the difference between monozygotic and dizygotic pairs. Alcoholism in monozygotic and dizygotic twins. There is yes number of twin pairs, monozygotic percentages, and dizygotic percentages. Adoption studies. Offspring of normal biological parents paired by schizophrenic adopting parents. Offspring of normal biological parents paired by normal adopting parents. Offspring of schizophrenic biological parents paired by normal adopting parents. The rate of schizophrenia in the biologic relatives of adoptees was shown to be 5%. Schizophrenia in biologic and adoptive relatives of adoptees who became schizophrenic. Here is the table. Migrant studies. These data strongly suggest that a significant environmental component is involved. Cancer of the stomach in Japanese men, 50, Missy, and US white. There is the growth of people and standardized mortality rate. Genetic technology. The Human Genome Project, use of genetic markers, gene expression, PCR methods, and genome-wide appreciation studies. Human Genome Project. Goal of Human Genome Project was to generate high-quality reference sequence of the base pair and identifying all humans. Become possible to carry out detailed sequencing of many individuals with certain diseases, family history, and environmental exposure. Genetic markers. Genetic markers are genes or DNA sequence that can be evaluated by laboratory. Single nucleotide polymorphism. SNPs are type of polymorphism involving variation of a single base pair. SNP correlates with disease and persistence. Second approach for studying genetic disease in laboratory is analysis of gene products, mainly proteins. Genotype to phenotype. Gene, gene products, functional and structural effects, symptoms and signs. DNA analysis, biochemical testing, and clinical evaluation. Gene expression is regulated by both on and off switch. By studying the kinds and amount of messenger RNA produced by cell, we can determine which genes are expressed. PCR method is a method widely used to rapidly make millions to billions of copies of a specific DNA sample, allowing scientists to take a very small sample of DNA and amplify it to a large enough amount to study in detail. PCR method. PCR implies two main reagents: primers, which are short single-stranded DNA fragments known as oligonucleotides that are a complementary sequence to the target DNA region, and the other is DNA polymerase. Basic steps in PCR methods are denaturation, annealing, and extension. Steps in PCR method. In the first step of PCR, the two strands of DNA double helix are physically separated at a high temperature in a process called nucleic acid denucleation. In the second step, the temperature is lowered and the primers bind to the complementary sequences of DNA. The two DNA strands then become templates for DNA polymerase to enzymatically assemble a new DNA strand from three nucleotides, the building blocks of DNA. PCR progresses, the DNA generated is itself used as a template for replication, setting in motion a chain reaction in which the original DNA template is exponentially amplified. Advantages and disadvantages in PCR method. Simple to understand and to use, produces results rapidly. The technique is highly sensitive with the potential to produce millions to billions of copies of a specific product for sequencing, cloning, and analysis. A real-time PCR shares the same advantages as the PCR with an added advantage of quantification of synthesized products. Therefore, it has its uses to analyze alterations of gene expression levels in tumors and microbes. Disadvantages: PCR users must know the precise sequence. Like all enzymes, DNA polymerases are also prone to error, which in turn causes mutations in the PCR fragments that are generated. Another limitation of PCR is that even the smallest amount of contaminating DNA can be amplified, resulting in misleading or ambiguous results. Single nucleotide polymorphism. In genetics is a substitution of single nucleotide at a specific position in the genome that is present in a sufficiently large fraction of the population. Single nucleotide polymorphism pinpoints differences in our susceptibility to a wide range of diseases like sickle cell anemia, beta thalassemia, and cystic fibrosis. Applications of single nucleotide polymorphism. Association studies. Tag single nucleotide polymorphisms are useful in whole genome single nucleotide polymorphism association studies. Haplotype mapping, disequilibrium linkage. Genotype versus phenotype. Genotype is the genetic makeup of an organism, whereas phenotype is the morphology, properties, and behavior of an organism. Genotype can be determined by observing DNA by genotype methods, whereas phenotype can be determined by observing outward factors. Genotype is completely dependent on the gene sequence, whereas phenotype depends on the genotype and environmental factors. Genotype is inherited by offspring, whereas phenotype is not inherited by offspring. Genome-wide association studies change the overall approach for studying association between genetic markers and disease. In genome-wide association studies using 1 to 5 million SNPs and lipids of their genomes strongly associated with disease. Focus on HLA types which are genetically determined, ankylosing spondylitis, pathogenic mechanisms involved, identify subset of population who are at risk, applying molecular biologic methods to family studies, determine the major identifiable gene transmitted from parent to child. It is associated with increased risk of disease. Segregation analysis observes pattern of disease in families and its compatibility with Mendelian mode of inheritance. Linkage analysis alleles from two loci segregate together in family and are passed as a unique unit from parent to child. Identify and isolate the genes associated with susceptibility to the disease pathogenesis, the development of appropriate preventive strategies. Confounder. Ancestry can be associated with exposure and with the disease. Simple stratification by self-reported ancestry, estimating the percentage of each person ancestry based on genetic material, selecting appropriate countries. Sequencing technology. The advancement in massive parallel or next generation sequencing technology becoming affordable even for sample sampling. Vast analysis for many complex diseases across large scale. Prospects for the future. Integration of knowledge of all these divergent areas may provide the foundation for earlier detection of high-risk individuals and could lead to more effective measure of prevention in coming years. Concept of causation, germ theory of disease. Lewis Pasteur demonstrated the presence of bacteria in air in 1860, whereas Robert Koch showed that anthrax is caused by a bacteria in 1877. Lewis Pasteur experiment to refute the theory of spontaneous generation. Broth in frost is boiled to kill pre-existing microorganisms. As frost cools, condensing water collects, sealing the mouth of the frost. If the neck is later broken off, outside air can carry microorganisms into birth. Here are the Koch's postulates. Isolate the pathogen from sick creature, grow the pathogen in the laboratory and obtain a pure culture, inoculate a healthy creature with the samples from the pure culture. The pathogen could cause the same disease symptoms that were seen in such creature. Re-isolate the same pathogen from the setting sequence. Germ theory of disease. One-to-one relationship between causal agent and disease. Disease agent enters man and results in disease. Epidemiological trials. According to this concept of disease causation, in addition to the causal agents, the factors relating to the host and environment are equally important to determine whether or not disease will occur in the exposed field. For example, not everyone exposed to Tubercle bacilli develops tuberculosis, but the same exposure in an undernourished or immunocompromised person may result in clinical disease and exposures occur more in overcrowding. This is epidemiological triage, environment, agent, and host interaction results in disease. Agent factors. Agent is defined as a substance, living or non-living, or a force tangible or intangible, the excessive presence or relative lack of which may initiate or perpetuate the disease process. A disease may have a single agent, a number of independent alternative agents, or a complex of two or more factors whose combined presence is essential for development of disease. Agent factors, biologic agents. These are living agents of disease, for example, viruses, bacteria, fungi, protozoa. Infectivity, ability of agent to invade and multiply and host. Pathogenicity, ability to induce clinically apparent illness. Virulence, proportion of clinical cases resulting in severe clinical manifestations including sequelae. Agent factors, nutrient agents, for example, proteins, fats, carbohydrates, vitamins, minerals, and water. Physical agents, for example, excessive heat, cold, radiation, electricity, sound. Chemical agents, endogenous. For example, urea, serum bilirubin, uric acid. Exogenous allergens, metals, fumes, dust, etc. Mechanical agents, exposure to chronic friction and pressure. Agent factors, absence or insufficiency or excess of a factor in facility health. Chemical factors, hormones or enzymes, lack of part of structure, chromosomal factors, Turner syndrome, immunological factors, for example, anemia. Social factors, for example, poverty, alcohol, and drug abuse, smoking, social isolation, etc. Host factors. In epidemiological terminology, the human host is referred to as soil and disease agent as seed. Intrinsic factors that influence an individual's exposure, susceptibility, or response to a causative agent. In some situations, host factors play a major role in determining the outcome of an individual's exposure to infection, for example, tuberculosis. Host factors, demographic, age, sex, ethnicity, biological, genetic factors, blood groups and enzymes, immunological factors, social and economical, socioeconomic status, education, occupation, lifestyle factors, use of alcohol, drugs and smoking, physical exercise and nutrition. Environmental factors. The external or macro environment is defined as all that which is external to the individual human host, living and non-living, and with which he is in constant interaction. Physical, non-living things and physical factors, for example, air, water, soil, radiation, heat, etc. Biological, living things around man, including man himself, for example, insects, rodents, animals, etc. Psychosocial include factors that stem from psychological makeup of individuals and structure and function of social groups. Triangle of epidemiology. In addition to host, agent, and environment, one more factor, time factor, is added. Time accounts for incubation period, life expectancy of the host or pathogens, duration of course of illness, environment, agent and host. Web of causation, suggested by McMahon and Smith. Ideally suited in the study of chronic disease where the agent is often not known and disease is the outcome of interaction of multiple factors. This model of disease causation considers all predisposing factors of any type and their complex interrelationships with each other. Here is the web of causation, and these are recent advances. Epidemiology is the branch of science that investigates the causes and distribution of disease in populations in order to provide preventive measures and promote human health. The fields of genetic and environmental